Amplification of gene of interest using PCR. Amplification of gene of interest using PCR. Amplification, it means to amplify, to multiply the copies of the gene of interest, while PCR is a method. So, in PCR, as you see, in PCR, it stands for polymerase chain reaction. Its name itself says the polymerase. Polymerase. It's kind of polymerization. The enzyme used is the DNA polymerase. Polymerase chain reaction, where we form the multiple copies of the gene of interest or DNA. This process is in vitro, takes place outside in lab. So in vitro. Now, using two sets of primers. Using two sets of primers. Now these primers are oligonucleotides that are complementary to the region of DNA and the enzyme DNA polymerase. The function of this enzyme, it extends the primers. In PCR, that is polymerase chain reaction. See in here, it's a polymerase chain reaction. 1 billion copies are made after 30 cycles. You see in the diagram, it says 30 cycles gives amplified 1 billion times. Now, this PCR, which is used for amplification of gene of interest, has 3 steps. The first one is denaturation. Second, primer annealing. As we already seen that 2 primers are needed. It's given that 2 primers are needed. In vitro process, 2 primers are needed, and the enzyme DNA polymerase that extends the primers. So this step, that is the primer annealing. That means it is binding of primers to the DNA strands. You see, initially in the first process, that is denaturation, 2 strands are separated under the heat. Just look at the diagram. After the heat, 2 primers are separate, 2 strands are separated, right? Then the primers attach, that the primer annealing. So this process, this step is annealing. Now, next step is the extension. Now, once the primers annealing is done, respective 5' to 3' 3' to 5', then DNA polymerase This is an enzyme. Now, this enzyme is thermostable. That means it can sustain the high heat. It can sustain the high heat. So thermostable DNA polymerase, and this isolated from a bacterium named Thermus aquaticus. It says which remain active during the high temperature, and that high temperature induce the process denaturation of double stranded DNA, because in PCR heat is required. Now this heat is required, it means the enzyme should sustain the high temperature, and that came from the Thermus aquaticus. Okay, in first step you see heat is required, that is the denaturation. That means the enzyme needed should be tolerate to high temperature. Okay, it means other enzymes cannot be used in this process PCR. Now talk about the extinction. So in the extinction process, DNA polymerase, we have already talked, that is thermostable DNA isolated from bacteria, which is Thermus aquaticus. Now this enzyme, that is DNA polymerase, also known as Taq polymerase, because it obtained from Thermus aquaticus. So Taq polymerase. Okay. Plus deoxynucleotides, which are the things, which are the components required for the synthesis of the strand, that is deoxynucleotides. So in extinction you see 5' to 3', 5' to 3'. DNA strand had opposite polarity. It means one strand is 5' to 3', the other strand synthesized will be 3' to 5', right? Opposite polarity. Okay, so these are formed in the extinction process. So in the result, after 30 cycles, this process continues, that is extinction, then denaturation, then annealing, then extinction, then denaturation. It gives 30 cycle amplified 1 billion times, right? So this was the PCR, that is polymerase chain reaction for amplification, that is the multiple copies of the desired gene or DNA or gene of interest. This is an in vitro process. Okay, you see the region to be amplified, it means a specific part of DNA, that is double stranded DNA, the region to be amplified, that is a gene of interest. So this was the process of PCR. Again I'm reading this in figure 9.6. PCR, that is polymerase chain reaction, each cycle, that means each cycle has 3 steps. First, denaturation, that separates the two strands of DNA. Then the annealing, that is the primer annealing. Primer gets attached, so the further strands generated. And then the extinction of primers. So primer annealing and the extinction of that primers. Okay, so this was the amplification of gene of interest using PCR, right? And remember this tag polymerase, no, Taq aquaticus, which is a bacteria, has thermostable DNA polymerase obtained. Okay, so this is the enzyme from Thermus aquaticus. This is why it is Taq polymerase, and it remains active during the high temperature that induced denaturation of double stranded of DNA. Okay, so it can remain active. So this is why this enzyme is good for this PCR process, because this PCR process involves the heat in the step in the first step. Okay,